Today we are going to study factors affecting enzyme activity. These factors include temperature, substrate concentration, pH, coenzymes, activators, inhibitors, water, and radiation. So let's start with temperature first. How temperature affects enzyme activity. Temperature, whether it is low or high, both can affect enzyme activity. Let me explain. How do they affect it? If the temperature is low, then enzyme activity decreases, but the enzyme does not get destroyed. However, if the temperature is high, enzyme activity stops altogether and the enzyme gets destroyed. So, an optimum temperature is necessary for all enzyme activity. The normal optimum temperature is between 30 and 37 degrees Celsius. Now let's move on to substrate concentration and how it affects enzyme activity. The concentration of the substrate is directly proportional to the rate of the reaction. This means if the substrate concentration is low, then the rate of reaction will also be low. But if the substrate concentration is high, then the rate of reaction will also be high. Now we come to pH. Just as an optimum temperature is necessary for normal enzyme activity, similarly, an optimum pH is necessary for enzyme activity. If the pH changes, the enzyme activity is affected. Either it gets destroyed or loses its activity. Did you know that every enzyme has a constant optimum pH? For example, the optimum pH for the pepsin enzyme is 1.6 and for trypsin it is 8.2. Now let's talk about activators. Activators are substances that increase enzyme activity, and each enzyme has its own activator. For example, magnesium is an activator for the phosphatase enzyme, and zinc is an activator for the carbonic anhydrase enzyme. Now we come to water. I hope that you all know how important water is. That is why our bodies consist of 70% water. Water also helps in seed germination, allowing the seed to grow. Now let's discuss radiation. Radiation such as ultraviolet radiation, alpha rays, beta rays, and gamma rays can destroy enzyme activity. Radiation will affect enzyme functions. Finally, let's discuss inhibitors. Inhibitors are substances that decrease enzyme activity. There are two types of inhibitors. One is competitive inhibitors, and the other is non-competitive inhibitors. I will be telling you both types. What are competitive inhibitors? The inhibitors that resemble the substrate are called competitive inhibitors. What are non-competitive inhibitors? The inhibitors that do not resemble the substrate are called non-competitive inhibitors. Additionally, competitive inhibitors fit into the enzyme's active site, while non-competitive inhibitors attach to the allosteric site of enzymes and alter the shape of enzymes so that substrate cannot fit into the active site. To make it summarize, in both cases, enzyme activity decreases. How does this happen? It will become clearer through the enzyme mechanism. I have created an animation to make it better understood here. You can see the enzyme here, and this site is the enzyme's active site. The site you see below or opposite the active site is the enzyme's allosteric site. To proceed further, let me give you a little definition of active site and allosteric site. What is an active site? The active site is where the substrate attaches, and the allosteric site is the opposite site where a non-competitive inhibitor attaches. This orange structure here is the substrate, and this is the competitive inhibitor, which resembles the substrate. And this is the non-competitive inhibitor, which fits into the allosteric site. Let me define all three again. What is a substrate? The substrate is the substance that fits into the enzyme's active site. What is the competitive inhibitor? The competitive inhibitor resembles the substrate and attaches to the active site, stopping enzyme activity. What is the non-competitive inhibitor? The non-competitive inhibitor attaches to the allosteric site and also stops enzyme activity. Now, How do these three work in the mechanism? Let's begin the mechanism with animation. Look at this. This is the enzyme, and this is its substrate. The substrate fits into the active site of the enzyme, forming the enzyme substrate complex, and the reaction takes place, producing the enzyme's products. Now let's look at how competitive inhibition occurs. Here is the enzyme, and here is the substrate. This is the competitive inhibitor, which fits into the active site, preventing the substrate from fitting in. That's why enzyme activity stops, or inhibition occurs, and this is called competitive inhibition. The reaction stops and cannot proceed further. Now let's discuss non-competitive inhibition. How does non-competitive inhibition occur? Here is the enzyme, and here is the substrate. This black structure here is the non-competitive inhibitor, which fits into the allosteric site. As soon as it fits, the shape of the enzyme changes, which prevents the substrate from fitting into the active site. Since it cannot fit, enzyme activity stops. Inhibition occurs again, and this type of inhibition is called non-competitive inhibition, preventing the enzyme from proceeding further. Question of the day. What type of molecules decrease enzyme activity by blocking the active site? Only a genius can answer in the comments. If you would like to watch my other lectures on topics like the lungs, kidneys, liver, brain and eyes, visit my channel to view my other videos.